Today's lecture will be the continuation of the studies on the approaches and methods on the study of promoter analysis. First, I'm going to take you to the WebCT site to show you a link with some specific readings on this, and then we'll have a bit of lecturing, and then I'll show you a couple of papers that are useful to look at. And eventually, for those of you in this semester 2005 when I'm recording this, but in the future it will be posted, I'll have lectures dedicated just to the analysis of those manuscripts, those research articles, and the figures. So let's go to the website. And of course it's under course materials, the course content, the DNA. Let's see. Yeah, it must be this. No. Got the wrong one, sorry. Well, we can use this same link, the antigen-antibody interactions. Oh, well, that's not it. Oh, it's one of these. Here, making and using antibodies. I thought I had a link to this same site for the DNAs, but when we click on the DNA and genes, all right, forward, that is important. And promoter analysis, the goals. And this describes some of the functional assays of promoters, defining the structure of cis-acting sequences by a functional assay, reporter genes, defining the limits of cis-acting sequences, transfection, stable and transient, and some transfection procedures. And a bit more information. And then there's the structure function analysis of promoters as well listed here. So this is a good site to go and look over the material relevant to our studies on the promoter analysis. We talked about the sequence isolation, binding capabilities of looking at transcription factors, and we're transitioning over to the functional assay. So I found my notes from prior years, and I'll scan these and post them, so we'll have something that isn't PowerPoint. So the key points were the gene expression, the constitutive but regulated, modulated when on, and the inducible, off then on, which are developmental, cell cycle, and immediate early genes, the ones that we focused on. I guess we hadn't mentioned the developmental and the cell cycle examples of inducible genes. Then we talked about the promoter region, identification, isolation, sequencing. An example of the identification was getting a mouse gene to get the rat gene, then use the rat gene to get the rat promoter. So some examples of how things have been done. With the sequences, there was the response elements that we talked about before, regulatory elements. And of course now we're beginning to see the origination of this terminology. Consensus sequence, when we're first sequencing it and we're finding it repeatedly on the promoter regions of genes that can be activated. And then response elements, when we see that those are genes that respond to, that contain those consensus sequences, those elements of DNA in the promoter region, and respond under certain conditions. And then finally, we talk about them as regulatory elements when we have a better understanding of them. So there might certainly be known ones, as we had mentioned, that can use a list. There might be new ones that would be identified from the studies. And remember, we're talking about the upstream region, not the downstream region. The upstream region is the promoter region. So obviously, knowing the transcription start sites is important, the TATA box and the AUG. And then there's even a neuron-specific sequence close to the start site that are useful in identifying when you're making that transition in sequencing upstream that you're getting into the promoter region. And then studying the regulation, which is what we're going to talk about today, the transient transfection, using in vitro cell culture, neuronal cell lines or other cell lines, as we'll see later and talk about more. These, of course, would be neuronal specific. There can be non-neuronal ones. We'll look at looking at different lengths, the end deletion, point deletion, mutations, either point or multiple, that you can utilize to evaluate when you're doing the transfections, and the utilization of a reporter region. So here's an example where an analysis was performed, and they kept reducing the length of the promoter region that they were putting into cells and testing it, and the shortest construct still had significant neuronal specificity. So we'll talk about this in more detail, but just even reading that sentence in our mind already, we should be able to realize that some of these portions of the DNA that are important for cell-specific gene expression can be close to the start site or far away. In other examples, the far upstream promoter region will be related to the ability to be induced during development, and the downstream promoter region can typically be associated with constitutive expression and allows the basal levels of the expression. So there is this generality of farther upstream being related to the developmental and sometimes tissue-specific or cell-specific gene expression, and those closer or downstream on the promoter region, closer to zero, related to the constitutive expression. And more specifically, this was related to this article, 2 alternative promoters, direct neuronal specific expression of the rat microtubule associated protein 1B gene. Doesn't have much to do with neuroendocrinology, but it certainly does relate to the method. And this is in the Journal of Neuroscience, August 15th, 1996, volume 16, issue 16, pages 5026 through 5036. What I'm going to do just quickly in doing that review and overview of the next step of our discussion is take you to the UTSA library. Let me find that spot while you're making note of that article. And show you how to get to some of these journals. I hope the Journal of Neuroscience is on there. So let's switch over to the computer. And on the www.utsa.edu, there's a link to the library. And when you click on that, you get databases and articles. The EJournal Locator is the one that I like to go through to first. And just type in Journal of Neuroscience. We do have the link from several different sources. I'm going to go to the Elsevier site. It's 1995 to present, so 96 it should show up. If you are doing this from an off-campus computer, when you're on UTSA using a UTSA computer, it'll take you straight to the journals that we have the subscriptions for. If you're off-campus, it'll prompt you for your input for the proxy, and you put in your last name and your @ number, your student ID number, then it'll take you to the site. So that was 1616, huh? Up to volume 141. Ooh, that makes the paper seem old right there. So 16. The Journal of Neuroscience. Did I get the Journal of Neuroscience? Journal of Neuroscience Methods. Haha. Okay. So let's go to Journal of Neuroscience, the one that we really want. So here's, I'm going to do the one with the PDF and the abstracts, 1996. And that was August 15th. August 16, here it is. And then we scroll down. It was 5,026. Looking at the page numbers. So 5,026. So you can download the PDF directly from there. We're actually not supposed to load these journal articles on the WebCT. I may have some already there. We're supposed to go through the library subscription so you can download them as a student, but we're really not supposed to post them. So this is my reason for taking you through this, so that we take every step that we can to be compliant with the copyright regulations. So I'll always give you the reference for articles so you can download them to have to go over when we do the lecture specifically on any article. I won't be covering any today. We just want to do some general discussion first. So what we want to talk about are in vitro methods of the analysis of promoter regions. So in the introductory, the overview and review and overview that we just completed, I mentioned transfection. So transfection is when you have the desire to get a gene, at least the cDNA, into a cell so that you can get expression of that and ultimately have this would be the cDNA, and ultimately have the message and the protein synthesized within that cell. So you want transcription and translation to occur in that cell. So there are a variety of ways of doing this. This is called transfection. And the terminology originates from working with bacteria and here is an article, just a brief letter on transfection versus transformation, defining the terms. So in 1987, the first parasite was evaluated for introduction of functional plasmid DNA. So it's a method that's been around for a while at that level using the plasmid approach, but there's very early work extending back to the 1940s of understanding that DNA can move from organism to organism. So it really isn't anything new. And basically, if you're talking about eukaryotic cells, as we are, will be using the term transfection. The transformation is typically reserved for looking at the prokaryotic types of cells. So this, if you wanted to go and take a look at it, is from Parasitology Today, Volume 16, Number 9. I got it zoomed in too much. 2002. And the author concludes, of course, with saying, I recommend to call it what it is, transfection. So the term transfection is what we will be using. And there are two types of transfection. There's transient transfection and permanent transfection. And so obviously the difference here is that with permanent, we'll just contrast them by saying that with permanent, the cDNA of interest is obviously incorporated in the host genome or DNA, and therefore with replication is passed from generation to generation. And so there's a couple types of permanent that we'll talk about. Obviously transient, you get it into the cell, and whether it gets into the nucleus or in the cytoplasm, and the cDNA is transcribed and message is translated, isn't a great concern. You can get transcription and translation by getting that cDNA into the cells under investigation and perform studies on that. And of course, what we're focusing on is getting a promoter region of interest into the cell, and then focusing on the gene product. Can we get a functional response that is dependent upon the promoter region? But first, I want to talk about the two different types of permanent transfection. The first is immortalization, consistent with our discussion of the in vitro. So this is for in vitro. And if you've ever tried to do primary cell cultures where you harvest the cells and try to grow them up, you realize that it's very challenging, because you're only going to get so many cell cycles out of those cells, and so you may get 2 weeks or 3 weeks to grow the cells up, try to get a sufficient number, and then still have time left in those cells before they just go through their final stages of senescence and poop out on you to complete your study. And then to do another study, you've got to collect some fresh organs, harvest the cells from the tissue, and start all over again. With immortalization, you have basically a tumor type of a cell that just likes to grow and grow and grow and grow and grow and grow, and you can utilize mitogens to trigger them to grow, and then take away the mitogens and stop them in their cell cycle, supposedly at their terminally differentiated cell cycle, whatever that might mean in vitro, and then stabilize them and perform your tests on them. And you can freeze them back and pull them out and grow them up at any time. So it is an optimization, if you will, of this approach. A pretty typical way of immortalizing cells, although there are different ones, is using the simian virus 40 plasmid, a portion of the this viral DNA, to integrate your gene into the host genome. Sometimes human retroviral DNA is also used. We know that retroviruses, such as the herpes virus and the human immunodeficiency virus, which obviously is not used to immortalize cells, but those are two examples of retroviruses that have within them all the machinery to insert their DNA into the host. So these are a couple of examples of the types of viral DNA that can be used to insert your DNA into that and then kind of trigger it into becoming a permanent cell line. So we have permanently transfected permanent cell line. Now, not every cell line is immortalized and transfected with a gene of interest at the same time. Sometimes they are done in sequence. You get an immortalized cell line, and then you start sticking genes into it to add to its gene collection as you sustain the original, maybe SV40 immortalization. So I don't want you to think that whenever you hear of an immortalized or permanent cell line that is transfected with a gene of interest, sometimes they just use these viral DNAs to make it a permanent cell line so it's easier to work with, and then along the way you can use similar viral approaches to insert genes of interest to test them, and they sustain throughout the progeny of that cell line. The second, of course, is in vivo, and we'll just use the term transgenics for that. So we'll have a brief presentation on that method as well. Going over here to look for reporter genes. Yeah. So I'm going to switch to the computer for just a moment. I know you can't read it, but there's this portion on the reporter genes, and two are mentioned here, luciferase and chloramphenicol acetyltransferase. I knew it was abbreviated CAT, but for the life of me I couldn't remember what CAT stood for. So I'll just use the acronym CAT. Don't confuse it with CHAT, which is choline acetyltransferase. This is CAT, which is chloramphenicol acetyltransferase, and luciferase, a couple of different enzyme genes that can be used as reporter genes. So let's get back to our idea of the strategy. There's always strategy in design. So the strategy to design your transfection probe. So we have a promoter region of interest. We said it can be of whatever length, and we're going to manipulate that. So it may be really big, like 5,000 base pairs. And you can generate this after it's been sequenced, obviously, and then produce it synthetically. And then attach to that, or ligase to that, a reporter gene. So what we're doing is we're interested in studying the promoter region, not really the gene that's being expressed. So what we want is a gene product that's very easy to detect and quantitate. So we can have an easily detected and quantitated assessment of the effectiveness or the function of the promoter region. So once we have this, what are the strategies to investigate that promoter region? As we had said, you can utilize different lengths So since you're able to synthesize the promoter region because you've sequenced it already, you can perform studies called truncation. You can truncate it with an end deletion. So you truncate it from one end to the next, or you can use a point deletion, start taking chunks out somewhere in the middle, maybe just remove what you suspect is a consensus sequence altogether. Or obviously there can be mutations, point or multiple mutations. Obviously you'd want to be targeting your candidate consensus sequences, excuse me. So obviously, after you've transfected your cell line with your reporter gene, then you obviously have to ask the question, if I am interested in a certain consensus sequence or regulatory element on this promoter region, I have to make sure that this cell either expresses the transcription factor that targets it, or I have to co-express, co-transfect the transcription factor as well. So these can become quite complicated. It depends on really the question you're trying to ask and the cell line that you're working with. Obviously, if the cell has a constitutive expression of the transcription factor targeting your candidate regulatory element or response element, then that's obviously a better way to do it, because getting stable transient transfections can be quite challenging. But basically, once you have the transfection completed, then you would, remember we said there was CAT, which is an enzyme. There was luciferase, whoops, that's a C, which is also an enzyme. And then there's a newer one, a wide variety of them actually, but the green fluorescing protein, was another that recently was added, and now they have many different types of these fluorescing proteins from microbes. The genes are from microbes or plants, or luciferase we know is from the firefly. So there's a variety of biological sources for these enzymes or proteins, and they can give you different signals to monitor. So obviously for the first two, you have to quantitate the enzyme activity, and the CAT enzyme is usually a color reaction, so it's a typical type of color-dependent absorbance type of an assay. Whereas the luciferase, as we know, comes from the firefly, so it's going to emit light, so you're measuring light intensity through one means or another. You might use that light to precipitate the silver particles, so that you've got silver grains that you can use to quantitate or actually be able to measure the intensity of the light directly. Same thing for the green fluorescing protein. You don't have to add any substrates for the enzyme. You just have to excite it with the appropriate wavelength, and then it will emit as any fluorescing agent would do. So then you have to have a device that can quantitate the intensity of the fluorescence at the level of excitation that you expose it to. There's another one to add to the list, an older one, LAC-Z. Now also, the reporter genes can be other proteins, although more commonly these are being used. These are proteins. They are being used, but you can use other proteins and then just use the Western blot procedure to look at the protein. So the Western blot is an approach for quantitating protein using antibodies, using immunochemistry. And we have a separate discussion of that. And of course now with all the approaches for assessing RNA, the messenger RNA levels for any of these reporter genes can be measured if a laboratory more readily performs the RNA analyses routinely than any of these others. So the reporter gene is not so much therefore focusing, or thereby focusing on gene product as it is on the messenger RNA. Anything to get a quantitative assessment of the effectiveness of the promoter region that you're evaluating. So let's say you you don't know if promoter region has any regulatory response elements, consensus sequences. As we said, the libraries were not always so available. So what do? You can do the deletions, like we were saying. You can truncate. So with the 5' deletions, you can find the shortest sequence that will still work. So obviously, if your promoter region was the 5,000, like we said, you just start knocking off 100 at a time. So 4,900, or whatever amount it might be, 4,800, drop down to 4,000. However refined you want to be in truncating the length of the promoter region that you transfect into the cell. And then you're using the reporter gene on all of these, obviously. So when the level of detectability of the promoter region disappears, say at this point here, you can detect your reporter gene, and it is not detected down here, then you know the consensus sequence. And as I said, you've got to be sure that there's just a particular goal that you're after. Maybe it's the cell-specific gene expression or a particular transcription factor that you know is produced at a high level in the cell line that you select, or you have co-transfected to overexpress a particular transcription factor. Whatever the strategy is, when you hit the point where you suspect that the consensus sequence might be located, and you clip that off and transfect with a shorter sequence so your reporter gene is no longer detected, then you know somewhere between 5,000 and 4,900 base pairs, so you only have 100 base pairs left to start playing with. So then you can start focusing on that. But if you did know or even after that point, you can begin to do the site-directed mutagenesis, where you just mutate the different regions. This may be just the 100 base pairs with the rest of the promoter region on there. And when you hit the site that no longer, or maybe it'll just drop off significantly, but there'll be a significant change, if not loss of detection of your reporter gene, then you know what was important. If it's a candidate for a new consensus sequence, then you can determine what parts of the sequence are very important. And through this as well, again, we're having the basic assumption that you are looking for a new consensus sequence. You replace or delete the individual bases throughout the area that you identified, and then you determine that it would be a consensus sequence. And then you start finding the proteins that can bind to that. So as we said, if you don't know what the transcription factor is, maybe you're studying the cell-specific gene expression, you just keep truncating and mutating until you found the portion of the promoter region that was important for the cell-specific expression of that gene within those particular types of cells. Then you use that sequence to start looking for the proteins that can bind to it. And then once you get the proteins, you can sequence those and identify new transcription factors that might be important in the cell-specific gene expression. Like the PIT1 was one that we mentioned in the anterior pituitary for the cell-specific gene expression. I guess I should have had this earlier in my notes. Other than the viruses for getting your gene of interest, whether it's in our case it's a reporter gene with the promoter region on it, there are other methods for transfection. There's a way of using calcium phosphate to precipitate the DNA. And then in the presence of calcium in the cell mixture, ultimately the DNA will get across the membrane and into the cell. Not very useful in eukaryotes as electroporation. Works quite well for bacteria. You just zap them with a little bit of electricity and you get the pores in the membrane and it'll close back up. There are detergent DNA complexes that can be used, or actual microinjection. Obviously, that's pretty labor intensive. More and more commonly, we're seeing the virus vectors being used to deliver, whether it's transient transfection or attempts for permanent transfection or stable transfection. So let's close by just talking about some limitations of this method, which is what we should always think about. So there should be some key questions, especially for the co-transfections that we would talk about. So there are really three difficulties. The first is has to do with the level of transfection efficiency. zoom out. So this will differ with different cell types. The purity of the plasmid. Again, we're thinking of the plasmid that contains the cDNA. So the purity of the plasmid preparations, abbreviated as PREPs there. The state of the cell used, the stage of the cell cycle or the phase of the cell cycle that they're in when you try to get the DNA in there, and a variety of other small details that we're not going to get into. So it's really quite a complex method, especially for verifying and validating that you have a nice stable transfection, even if it is transient, and that you have a good efficiency. The second is the efficiency of the plasmid introduction. Into a cell in the first place. So it's not of the highest efficiency. It's usually only a few percent of the copies of the plasmids that you start with that actually will get into the cell. Sometimes it'll be a small number of cells that will take it up. So you have to be able to tell, to select the cells that have it in there. And of course, there are limits of sensitivity. So how many copies do you get into each cell? How many cells of those that you expose do you get it into? And then are they all collectively at a level that falls well within your methodological sensitivity range to detect that you've got a successful transfection in the first place before you begin your experiment? Or the third and final one, the introduced DNA is just lost. Remember what episomal means, but that was the explanation used. So it's treated as episomal DNA and just destroyed. So finally, just to clarify, I've been using stable and permanent a couple of different ways, and let's just close with that then. So the two approaches were what I said before was permanent. It's the stable transfection. And that's the one where it is integrated and goes on with the cell cycle. And then I know just a moment ago I used the term stable transfection with the transient transfection. And what I was trying to point out was that your transient transfection stayed stable over a period of time. So typically you have an opportunity to study the promoter region and the reporter gene within 24 to 72 hours with the transient transfection. So that's what I was saying, is that how stable is it? Can you get the expression to a good level, well within your sensitivity range, so you can determine decreases and increases If you just barely get it to the level of detectability with your method, and that just barely within your sensitivity range, you can't get ups and downs. You can only get increases above your baseline expression. It's very important to establish the efficiency when there is co-transfections. And sometimes there'll be co-transfections just to follow the co-transfected gene in with your gene of interest, so you can get a ratio that indicates the efficiency. So you may see in articles that you read, they'll say that the cells were co-transfected with the promoter region for growth hormone receptor and CAT or something else. Whatever they're co-transfecting with, they'll measure that in the cells, and also a level of their particular gene or reporter gene on the cDNA of interest, the one that they really want to study. And if they get a good ratio, a high ratio, almost a 1-to-1 in the level of expression of each of the co-transfected genes, then they feel that they have a very efficient transfection. So when we look at a couple of papers or ultimately talk about methods, the utilization of these methods to study promoter regions, it'll seem very simple when we hear it. From this discussion, we want to be able to understand that it isn't so simple, and we need to be critical in evaluating that type of information. So I'll stop there, and we'll have one more session like this where I'll discuss the transgenics just briefly, and we'll go on to other methodological approaches. So I appreciate your time and attention, and hope to see you all again soon.